Hello, I'm Richard Young, your host with Facundo Batista and Lena Fayan for MIT's course on COVID-19, SARS-CoV-2, and the pandemic. Yesterday, Pfizer announced that its RNA vaccine against COVID-19 is highly efficacious, exceeding expectations and providing hope that the pandemic will come to an end in the foreseeable future. Today, we have the pleasure of hearing from Dr. Kizzmekia Corbett, who's a research fellow and the scientific lead for the coronavirus vaccines and immunopathogenesis team at the National Institutes of Health Vaccine Research Center, and who has been working on a very similar RNA vaccine against COVID-19. This class of vaccines is new and exciting. They can be developed quickly and bode well for our response to future pandemics. Dr. Corbett is a viral immunologist and obtained her PhD in microbiology and immunology at the University of North Carolina at Chapel Hill in 2014. She then joined the NIH's Vaccine Research Center and worked on development of a universal influenza vaccine, which will soon be in clinical trials. Dr. Corbett's work now focuses on developing vaccines against the coronaviruses. This includes mRNA 1273, developed in a partnership between NIH and the biotechnology company Moderna. This vaccine is currently in a phase 3 trial, the final phase to determine its efficacy against COVID-19. Dr. Corbett, thank you so much for joining us today.
Absolutely. Thank you for the kind invitation. I am very happy to be sharing some of this work, particularly, as you said, with the news that came out yesterday around Pfizer's mRNA vaccine candidate. So I generally give very, very specific talks around our particular vaccine candidate that we're co-developing with Moderna, mRNA 1273. But I wanted to start this talk because it is to a more broad audience with just a clipping that I got from YouTube around how vaccine development works and how the vaccine development process for COVID-19 has been so rapid.
Vaccine development can generally be split into 3 phases. In exploratory research, scientists experiment with different approaches to find safe and replicable vaccine designs. Once these are vetted in the lab, they enter clinical testing, where vaccines are evaluated for safety, efficacy, and side effects across a variety of populations. Finally, there's manufacturing, where vaccines are produced and distributed for public use. Under regular circumstances, this process takes an average of 15 to 20 years. But during a pandemic, researchers employ numerous strategies to move through each stage as quickly as possible. Exploratory research is perhaps the most flexible. The goal of this stage is to find a safe way to introduce our immune system to the virus or bacteria. This gives our body the information it needs to create antibodies capable of fighting a real infection. So the best way to speed things up is for many labs to work on different models simultaneously. This race to the finish strategy produced the first testable Zika vaccine in 7 months, and the first testable COVID-19 vaccine in just 42 days. Being testable doesn't mean these vaccines will be successful, but models that are deemed safe and easily replicable can move into clinical testing, while other labs continue exploring alternatives. Whether a testable vaccine is produced in 4 months or 4 years, the next stage is often the longest and most unpredictable stage of development. Clinical testing consists of 3 phases, each containing multiple trials. After a successful phase 3 trial, a national regulatory authority reviews the results and approves safe vaccines for manufacturing. Every vaccine has a unique blend of biological and chemical components that require a specialized pipeline to produce. To start production as soon as the vaccine is approved, manufacturing plans must be designed in parallel to research and testing. This requires constant coordination between labs and manufacturers, as well as the resources to adapt to sudden changes in vaccine design. Our best strategy is for labs around the world to cooperate and work in parallel on different approaches. By sharing knowledge and resources, scientists can divide and conquer any pathogen. Vaccine development.
So as that video stated, this entire vaccine effort around coronaviruses has been a really concerted effort with multiple labs across the country. The field is wide open, and there are 3 basic levels of vaccine development. Largely the preclinical testing phase, and then the clinical testing phase, and then the manufacturing phase. So I'm going to talk a lot today about the preclinical testing phase that has driven this rapid vaccine response across the field. So by way of introduction to coronaviruses, which if you are attending this meeting on a regular basis, you probably fairly well understand that coronaviruses are a large family of viruses that have pandemic potential. And so what does that mean? It means that these viruses that generally dwell in animal reservoir, just like about 23 other viral families, have the potential to jump from their animal reservoirs into humans. So this has been happening with coronaviruses. It started with the SARS epidemic over 10 years ago that tallied over 8,000 cases worldwide. And then the MERS epidemic that started in 2013 that has since tallied over 2,000 cases worldwide. So history does repeat itself, but also science generally should guide us. And with that, our close collaborator, Ralph Barrick's laboratory, published a series of papers stating that SARS-like viruses, like we have now in this pandemic, SARS-CoV-2, are poised for human emergence. And so we knew that these viruses were poised for human emergence, and the universe has now responded with a big ol' "I told you so." The 2019 human emergence of SARS-CoV-2 has now turned into a global pandemic that, as of about 2 nights ago when I made these slides, had tallied over 50 million cases worldwide. So here we are in a global pandemic that could really only be subsided with some level of high-level herd or community immunity. As you may very well know, in the United States, we have now hit our 3rd wave of coronavirus COVID-19 cases. This red blip here on the far right of this graph, starting in around mid-September, shows the cases starting to increase. And if we think about what is called herd immunity or population immunity, we need about 60 or 70% of that so that we can return back to normal activity. It is notable that even at a 1% case fatality rate, over 40 million people would succumb to COVID-19 globally, shall we let the virus continue. And thus, a vaccine is needed to prevent the dynamics of the global pandemic. And so what would a vaccine do? Well, firstly, on the individual level, a vaccine could reduce the severity of COVID-19 illness and ideally prevent infection. From a community standpoint, vaccines have the ability to reduce transmission and thus make it safer to resume our normal activities. What does it mean to have from a community standpoint, and what is population immunity? Well, if you take this graphic of these stick figures that are aptly colored in a non-immune population where all of the people are blue, if a virus is set free into that population, there will be high levels of transmission where people in red will get infected and some may even succumb to the viral infection. In an immune population, however, where some of the people are green, you can see that the green people do not have the ability to get infected with the coronavirus because they are immune. And what's interesting about this scenario also is that the red people that were not immune are somewhat protected because of their distance from between each other. And so there's this level of about 70% of that type of green immunity that is needed to really allow for us to get back to our normal activity. And so how do you really want to elicit herd immunity? The way that you probably most likely hear about it and the way that it got sort of a bad name is via media, where they were describing herd immunity being induced by infection. As I said, the outcomes of herd immunity being introduced by infection could potentially increase disease severity and the amount of transmission, obviously, and also there are variable antibody responses that people acquire following natural infection with SARS-CoV-2. And so we don't really understand how viral infection might elicit immunity. There's also vaccination. So I have here different types of vaccine platforms, whether it be introducing the body to a protein or introducing the body to a virus-like particle or a nanoparticle, or even gene-based delivery, which I will get into some detail today because that is the strategy for which we are using with our collaborators at Moderna. Nevertheless, no matter how you elicit or give a vaccine to people in a population, this is called rationally designed vaccines. And so it's rationally designed because you are giving a substance to a person, and that person will elicit an immune response to that particular protein or VLP or what have you. And that immune response will then prevent infection or disease later. Antibody responses can also be given directly. So you've probably heard about plasma therapy that is in phase 3 clinical trial right now as well. But the daughter, so to speak, of plasma therapy are monoclonal antibodies that I won't talk to you about today. But the beauty of monoclonal antibodies is that they can be used also prophylactically or therapeutically. The immune responses are being tested for several monoclonal antibodies as right now in phase 3 clinical trial. And so we know that we want to most effectively elicit population immunity by vaccine. And this was briefly stated in this particular
to
clip.
find treatments for the sick and a vaccine to protect the healthy.
This ends with most or all of us being immune to this virus, and ideally that's through a vaccine.
Can solutions that usually take years
we are trying to move as fast as we possibly can
be found in a matter of months?
It is an extremely revolutionary approach to get into a phase 1 clinical trial 66 days after the sequence of a virus is released is a world record.
And so how did we get to a so-called world record? The way that we were able to use SARS-CoV-2 vaccine development as a proof of concept around rapid vaccine development was by using what we call pandemic preparedness. The approach that we took, particularly in our laboratory, was the prototype pathogen approach that states that one might do research on a known virus within a given virus family. In the case of the coronavirus family, we chose the MERS coronavirus to use as our prototype pathogen, generate enough knowledge around that particular virus so that it could be applied widely to that viral family. And then ideally for a pandemic response, you would want to develop that countermeasure or vaccine through a phase 1 clinical trial. And while we did not do that in this particular case with MERS, we did have enough generalizable knowledge around MERS coronavirus vaccine development that allowed us to move fairly quickly in this vaccine response that benefited the entire rapid development of multiple vaccines. So as we think about new technologies for transforming vaccinology, we like to think about them in kind of 2 different bubbles. The first of those is precision. So this is utilizing technologies, structure-based vaccine design, mapping vaccine responses and immune responses, whether they be in the preclinical setting in animal models or in humans, defining antibody lineages, all of these things that really inform how to best make the vaccine antigen or the piece of the virus that you want to deliver to a person to elicit a vaccine response. And then there's speed. Speed is obviously important because we are witnessing that right now, where you do need to be able to monopolize on a vaccine platform that has the ability to be manufactured fairly rapidly. And generally speaking, those types of platforms are or mRNA type platforms or DNA type platforms are tending to transform vaccinology in that way. And so in our vaccine preparedness for the pandemic threat of coronaviruses, it was obvious that we needed a fast, as I discussed, vaccine technology to be able to be produced in vast quantities quickly, reliable, a vaccine technology that must be tested and uphold some level of manufacturability standard, And then italicized universal. If you came on this call prior to the course starting, you note that I was asked about universal influenza vaccines, for which I have some history in understanding and developing those as well. So the way that we think about universal as yet for coronaviruses is italicized because it is not necessarily how we think about universal influenza vaccines. But what I hope to show you today is that the idea around universal is more so around a plug and play method that might be preemptive that will allow you to move quickly into human phase clinical trial. And so how do we do this, and what was our timeline for development? And I'm particularly going to talk about the candidate, the mRNA 1273, that our laboratory is developing in collaboration with Moderna. But there are several candidates that have taken a fairly similar rapid timeline to vaccine development. As you are very well aware, the first report of a respiratory virus outbreak in Wuhan, China, occurred in the latter parts of 2019. By January the 10th, the sequences for what was then called 2019 N coronavirus were published online. And then only 66 days later, a phase 1 clinical trial with mRNA 1273, Moderna SARS-CoV-2 vaccine candidate, happened in what is so-called a world record to enter a phase 1 clinical trial. And how did we do that? Because we really spent time on the preclinical development of this vaccine in the anticipation of the pandemic, thinking about a prototype pathogen approach using MERS coronavirus and other coronaviruses. And so the pandemic, the prototype pathogen approach that we use utilize the vaccine target, which is the coronavirus spike protein. So on the left-hand side of your screen, you see a coronavirus that has this spike protein that is this trimeric protein that sits on the surface of coronaviruses. On the right-hand side of your screen, you see a human cell that has a human cell receptor. In the case of SARS-CoV-2, that cell receptor is called the ACE2 cell receptor. Binding of the spike protein to the human cell receptor occurs via a receptor binding domain that I've highlighted here. That receptor binding domain is housed in this head of the spike protein that is called the S1 region. And then below that, in the stalk or the S2 region, is where the fusion machinery lies. So that is a very important point. These spike proteins are called fusion proteins because their entire purpose is to allow for attachment to the human cell, but then also allow for the virus and the cell to fuse together, and so that infection and replication can occur. Fusion proteins, such as the coronavirus protein, have been used as vaccine antigens before, and the proof of concept around stabilizing these proteins in what's considered to be a pre-fusion conformation to improve vaccine potency came via the respiratory syncytial virus F protein. So if you draw your attention to the right-hand panel of this slide, where you see two respiratory syncytial virus F proteins, one of which is elongated and called the post-fusion conformation. The second of which is more squatted and called the pre-fusion conformation. Looking at the red surfaces on these proteins, these surfaces are where the neutralization sensitive epitopes lie. For lay terms, those are the surfaces that you want to present to the body and make sure that the body sees them so that the body makes really robust antibody responses. So if you present this post-fusion protein in its post-fusion conformation, those red surfaces are hidden and occluded, and so the body cannot see them. Whereas the pre-fusion protein has those red surfaces displayed via like show and tell to the body, and therefore, when you give a vaccine in the pre-fusion conformation, you can elicit more robust neutralizing antibody responses. So we wanted to do the similar kind of thing with coronavirus spike proteins. At the time, when I came into the Vaccine Research Center, there were no coronavirus spike structures that had been solved. And so we didn't really have intel on the types of detail that or immunogenicity that a spike protein might be able to elicit because we didn't know what it looked like. And therefore, in collaboration with Andrew Ward's laboratory and Jason McClellan's laboratory, we solved the first pre-fusion structure of human coronavirus HKU1 spike protein. At the same time, the Wiesler laboratory solved the structure of the MHV coronavirus spike protein. And then there was this real onslaught of literature where different laboratories were describing various coronavirus spike proteins and really trying to lay out the architecture of these spike proteins. Around the time of 2017, George Gao's laboratory published cryo-EM structures for MERS and coronavirus SARS coronavirus spike proteins. We also, at the same time, published the spike protein structures for MERS coronavirus, and hidden in that paper also some SARS and HKU1 as well. But you'll note that in our paper, we described the immunogenicity along with the structures of those spike proteins and termed the spike proteins not just some spike proteins, but rationally designed pre-fusion spike antigens, because we were really interested in the utility of these spike proteins as vaccine antigens. And so how do you rationally design a spike protein and lock it in its pre-fusion conformation? The first clue is to hone in on the part of the spike sequence that is where the fusion peptide lays. So this blue FP is the fusion peptide. That region of the spike protein is called the S2, is within the S2 region. And you can see that these coils look like the toys that you used to play with as a kid that spring up and down. And so to lock it down and without allowing that coil to spring on top of each other, we needed to insert mutations into the backbone of the spike protein. In collaboration with Jason McClellan's laboratory, namely with Ning-Chong Wang, who's no longer in that laboratory, but did a lot of this work, we scanned the spike protein for mutations that might be able to stabilize it in its pre-fusion conformation, stumbling upon, and I say that very loosely because it was several years of work, 2 proline mutations that effectively locked the protein into its pre-fusion conformation. And how do we know that that was happening? Well, firstly, the S2P, so-called, expressed 50-fold greater than the MERS S wild type. And also, when you look at these proteins under a microscope, what you see is that the S2P version is in its pre-fusion, mushroom shape, beautiful, homogeneous, while the S wild type version of the protein readily flips into its post-fusion conformation. And these 2 proline mutations had the ability to be transplanted into the backbone of other other coronaviruses as well, inclusive of HKU1 and also SARS-1 and several other ones. But most importantly is that we were able to effectively stabilize the potentially emerging WIV1 SARS coronavirus spike protein in its pre-fusion conformation with those 2P mutations. And so, by way of prototype pathogen design, or the prototype pathogen approach, we would wanted to test the immunogenicity, or how well do these proteins elicit immune responses starting in a small animal model. Here we use the BALB/c mouse, where we immunized at week 0 and week 3, and simply took a bleed 2 weeks after that immunization, comparing various different constructs. The first is the golden child, the MERS S2P construct in its pre-fusion conformation. And then the MERS S wild type, which readily flips into its post-fusion conformation. And then a MERS S1 monomer. And without belaboring the point of one of the reasons why we use that is because in previous approaches from our center, the S1 monomer elicited very robust neutralizing antibody responses. And so we wanted to really set that standard bar around eliciting antibody responses. And so in testing those vaccine candidates in mice, what we show is that in a dose-sparing way, MERS S2P elicited more robust autologous neutralization, or neutralization against a virus that is for the same strain for which the spike protein was taken. But it wasn't just for autologous neutralization, it was also for homologous neutralization against multiple MERS coronavirus strains. And so now we had this S2P protein that elicited very robust neutralizing antibody responses. But in the case of a pandemic, developing a protein would take some time because of just the way that these proteins have to be manufactured. And so we needed to use a vaccine platform that had the ability to be fast, produce quickly in vast quantities, was reliable, so at least tested in humans through phase 1 clinical trial, and also italicized universal in that you could plug and play the type of therapeutic protein that you wanted into the system and really pull the trigger on manufacturing. And so we did that. We, in collaboration with Moderna, which is a pharmaceutical company that uses messenger RNA to deliver drugs, inserted the MERS S2P, again, from our prototype pathogen, into the backbone of their messenger RNA vaccine platform, which consists of purified messenger RNA for the spike protein that is encapsulated in a lipid nanoparticle. That lipid nanoparticle is then delivered into a muscle cell, and the muscle cell reads that mRNA and then essentially spits out spike proteins, and the body produces an immune response to it. To test this as a proof of concept with MERS S2P, we delivered that protein via mRNA in an animal model, in a mouse model that had a human DPP-4 receptor. So this mouse had the ability to succumb to a lethal dose of MERS following challenge. You can see that in the animals that were given PBS only, that they succumbed after 8 days following the challenge. However, animals that were given 1 or 0.1 microgram of MERS S2P mRNA survived that challenge, and even at a very low dose of 0.01 microgram per mouse. While some animals did lose weight, the animals did start to recover and did not fully succumb to the infection. And not only was it via weight loss that we saw this protection, but also via the amount of virus that was replicating in the lung. Taking those lungs and looking at the amount of virus inside of them, we showed complete protection at the modest 1 and 0.1 micrograms of MERS S2P mRNA. Also looking at a disease score called lung hemorrhage. So this is just how much hemorrhage is inside of the lung, whereas 4 is a score with a lot of hemorrhage and 0 is no hemorrhage at all. There was no hemorrhage at all in the lungs of the animals that were immunized with 1 or 0.1 micrograms of MERS S2P mRNA. So what this showed us is that by proof of concept that MERS S2P mRNA could elicit dose-dependent protection against lethal MERS challenge in what's called 288-330 mice, or mice that have the human DPP-4 receptor. And so we were very confident in our ability to deliver spike proteins, particularly the S2P protein, via mRNA. And thus, once the sequences for SARS-CoV-2 came out on January 10th, in collaboration with Moderna, we were able to rapidly, in 3 days, decide on a sequence for mRNA 1273. The mRNA 1273 encompasses the SARS-CoV-2 spike protein anchored by its transmembrane region, so that each cell, the cells present the spike protein on the surface. And also we were able to show, in collaboration with Jason McClellan, using those 2 proline mutations, that we could effectively stabilize a secreted version of the spike protein, which led to the structure of the SARS-CoV-2 spike being published in 37 days. I want to just take a step back from the vaccine response to really hone in on all of that, what I've said about the protein, particularly because it's not just for vaccines that these high quality stabilized proteins are beneficial. I'm telling the story about a particular vaccine candidate being developed by Moderna, but then there's also the use of this particular protein with those proline mutations to isolate antibodies between our laboratory and a small biotech company in Canada called Accelera, where this particular very potent neutralizing antibody is being developed by Eli Lilly. And it was shown yesterday that it was being used, it can be used by emergency use authorization by the FDA. It was the first antibody to enter into phase 1 clinical trial in the United States. Also, all across the globe, I don't have a very recent count, but there were hundreds of diagnostic tests being developed with this particular SARS-CoV-2 S2P protein. And it is of important note that 5 out of the 6 leading vaccine candidates that are currently in the pipeline also used the S2P mutation to stabilize their vaccine antigen despite whatever the backbone of their platform is, including the Pfizer BioNTech vaccine as well. And so when we decided on the mRNA 1273 sequence, it was also only 37 days that we were able to confirm that the vaccine was eliciting animal immune responses in mice. And the way that we confirmed that is by immediately, when we decided on the sequence, and got the vaccine from Moderna, we immunized 3 different strains of mice, given the standard 2-dose vaccine regimen and testing the immune responses both after the first dose and also the second dose. The gist of these graphs is that you elicit a potent antibody response following 1 dose that is boosted following a second dose. And it wasn't just that the antibodies bind to the spike protein, but also, and particularly at the 1 microgram, which is a very modest dose, we elicit neutralizing antibody responses. But that's after 2 doses. What happens after 1 dose? After 1 dose, you can see following 2 weeks and then 4 weeks following 1 dose, that 10 micrograms and 1 micrograms elicits about 10 to the 3 IC50 neutralizing antibody titer. So we're giving these fairly modest doses of mRNA 1273, and we're eliciting neutralizing antibody responses that are robust and, based on what we understood about MERS, should be protective. Therefore, we wanted to test whether or not there was this vaccine was effective at protecting against SARS-CoV-2 in a mouse model. And so again, in collaboration with Ralph Barrick's laboratory at the University of North Carolina, we used their mouse-adapted strain of SARS-CoV-2. So this is a virus that they tweaked in the receptor binding domain so that it binds to a mouse ACE2 receptor, and then upon binding to the ACE2 receptor in a mouse, has the ability to replicate in the lung and also in the nose. And so we tested for whether or not our vaccine, mRNA 1273, could decrease replication in both the lung and the nose. And as you can see, at 1 micrograms, we do not see any viral replication inside of the inside of the lung. And then in a dose-bearing way, you see that there is viral replication and breakthrough starting at the 0.1 microgram dose. Also, in the nasal turbinates, 3 out of 4 of the animals that were harvested at this time point did not show any viral replication in the nose. But that's after 2 doses, and what about after 1? So 7 weeks following 1 dose of mRNA 1273, we were able to show that mRNA 1273 protected after a 10 microgram dose or a 1 microgram dose in the lung. With that being said, that was very interesting data, particularly as you think about the utility of vaccines in an outbreak setting. So we moved our vaccine test up into a higher order animal, where we now are testing in rhesus macaques, giving the 0 and 4 week immunization, and then testing whether or not there's protection following challenge with about 10 to the 6 PFU of SARS-CoV-2, 4 weeks following the second dose. What you'll see in the slides following are 2 doses of mRNA 1273, the first of which is a 100 microgram dose that is quote unquote clinically relevant, and the dose that was moved forward into phase 3 clinical trial, and then 10-fold less at a 10 microgram dose. We looked at antibody and T cell responses and protection in the upper and lower airway. I don't want to belabor the antibody responses or the temporal antibody responses here. I want to really point towards the neutralizing antibody responses, where obviously before vaccination, we do not have any antibodies to SARS-CoV-2. Following 1 vaccination, you see this boost in antibody response that holds still until the 4 weeks following the first vaccination. Then with a boost of mRNA 1273, there's an increase in the neutralizing antibody response, where we get about a 10 to the 3, 10 to the 4 neutralizing antibody response that holds steady at least through 4 weeks following the second vaccination. Taking this time point, I want to show you some data from a couple of functional assays. So this is the ACE2 binding inhibition at week 4 following the second vaccination, where we are really just testing the amount of antibody that has the ability to block ACE2 binding from the receptor binding domain. There are several points to take away from this graph. The first of which is that we tested against a large panel of serum that's collected from people that are naturally infected, and we're able to show that we elicit antibody responses that are above that of people that have been naturally infected and recovered from COVID-19 disease. And this is a wide range of disease, severe infections, et cetera. It was very interesting to note that even with these, what we thought would be a subprotective dose, but 10 microgram dose of mRNA 1273, we were still eliciting a fairly robust ACE2 binding specific inhibition antibody response. And we looked at our live virus neutralizing antibody titers, because what I've shown you previously have been pseudovirus responses, and the gold standard in the field is to really look at live virus neutralizing antibodies to detect how a vaccine response might impede not just viral entry and attachment, but also replication. And so in this particular assay, which is a nanoluke assay developed by Ralph Barrick's laboratory, you can see that there's whopping 10 to the 3 live virus neutralizing antibody titers that far exceed that of convalescent sera. As you can tell here, there's about 1/3 of people that actually fall out of having neutralizing antibody responses following natural infection. So to see 100% of the animals being above that was very exciting to us. We also looked at T-cell responses, mostly because T-cell responses can be correlated with safety signals, particularly for respiratory virus vaccines. Some data that have from previous vaccines show that if you elicit a response that is more skewed towards Th1 versus Th2, you have you're less likely to elicit a vaccine-induced enhancement response. Not to belabor the T-cell responses at all, but only to show that 7 out of 7 of the animals that got the 100 microgram dose of mRNA 1273 do have a Th1 CD4 response, whereas only 2 of them have a Th2 response. And as you can see, it's really abysmal and very low level Th2 response. So we do have this skewed towards Th1 as we would like. We're also eliciting TFH cells in these animals, which is in line with what you would say about, and what you would expect, in regarding what we know about our antibody responses. Looking at the ability of the virus to replicate in these animals that have been vaccinated, both in the lower airway and the upper airway. The assay that we're doing here is subgenomic RNA assay that really detects nascent viral replication. And you can see that in the lower airway, 100 micrograms or even 10 micrograms is very well protective against rapid clearance of viral replication. And then also what was very, very, very cool for us to see, particularly at this time where no other vaccine in the literature had shown robust protection in the upper airway via nasal swab, we were able to show with a 100 microgram dose of mRNA 1273 that by day 2, all of the animals had cleared the virus from the nose. And so what that signified to us is that not only does this vaccine have implications around protection of disease, but there might be some implications around transmission as well. And so on the other side of the spectrum, so there's the preclinical side of things, and then there's also the clinical side of things. In the general vaccine development process, there would be some stepwise order to that. But in this vaccine process, because there needed to be a vaccine developed very rapidly, things were happening synonymously and almost at risk. So at the same time that we decided on the sequences for mRNA 1273, Moderna pulled the trigger on GMP production, or production of clinical grade vaccine, so that we were able to go into phase 1 clinical trial very quickly, even without having seen any of the animal responses. Now, obviously, the FDA wants to see the animal responses, but we were not willing to wait that month before getting that data in order to start manufacturing. With that, Moderna was able to ship the clinical drug product 41 days after they started their production, and then the phase 1 clinical trial started soon after. I am going to summarize just very briefly for you because I'm running out of time. The phase 1 clinical trials and then some of the vaccine landscape just in general for coronavirus. So there were 2 phase 1 clinical trials for mRNA 1273. The first of which, that one that started in Seattle in 66 days, was with people aged 18 to 55, looking at 3 different doses of mRNA 1273. And then in the second trial, there was an addition of people who were over the age of 56, looking at 25 and 100 micrograms of mRNA 1273. These volunteers got 2 doses of mRNA 1273, and they will be followed for a year. But the data that I'm going to show you today is going to be for antibody responses that are out to 57 days within the trial. So these are just some of the data from the preliminary reports. So we are eliciting very robust S-specific antibody responses, as you can see, after the first dose, and then a boost of about a log of the of the endpoint titer for S-specific antibodies after a second dose. Those antibodies are notably not waning through day 57, and also notably in the upper quartile of what you see from in human convalescent sera. Looking at the neutralizing antibody responses via our standard pseudovirus neutralization assay, there are a couple of things that are of note. The first of which is that you don't get 100% of vaccine neutralizing antibody responses until after a second dose, particularly in this and even in the 100 microgram group. But after a second dose of mRNA 1273, you get neutralizing antibody responses that are also in the upper quartile of what you see for human convalescent sera. And while those were the younger adults that were enrolled in the first clinical trial, comparing their neutralizing antibody responses to people that are from age 56 to 70 and greater than 71, you can see that the older adults elicit just as robust neutralizing antibody responses as younger adults, which has some promise because as you think about vaccines, they might not, generally speaking, vaccines may not have the same type of immune response in older people, and the immune response sometimes is dampened in older populations. And so in conclusion, we were able to rapidly develop a vaccine using 2 proline mutations to stabilize the spike protein in its pre-fusion conformation. And a large body of work around a prototype pathogen MERS coronavirus was able to inform how we moved forward with this vaccine candidate and delivering it by messenger RNA in collaboration with Moderna. And the vaccine candidate elicits, is highly immunogenic and protective in animal models, and also elicits robust immune responses in humans. And as of today, the vaccine candidate is in phase 3 clinical trial that has completely enrolled 30,000 people. Now, to give you just a slight and brief overview of the SARS-CoV-2 vaccine pipeline across the globe. There are over 200 vaccines that are at least in preclinical testing, but I will focus on the vaccines that are in human trial. So there are 36 vaccines that are in phase 1 clinical trial. So this is just to assess safety and dosage. Expanded phase 2 clinical trials where additional safety analyses occur and you try to pinpoint your dose for your vaccine. There are 14 vaccines that are in phase 2 clinical trial. And then there are 11 vaccines that are in phase 3 clinical trial. And these are the large scale vaccine trials that assess efficacy. And there are 6 vaccines around the world, not in the United States, though, that are approved for early or limited use. And no vaccine to date has been approved for full use. Focusing in on those vaccines, particularly those that are in phase 3 clinical trial in the United States, there's the Moderna candidate, which I told you about throughout this presentation. Then there's the Pfizer-BioNTech candidate, which is very similar to the Moderna candidate in that it is a pre-fusion stabilized spike that is being delivered by mRNA and lipid nanoparticle. They are planning to enroll 40,000 people in the US and Brazil and have recently expanded their trial to children as well. There's the Janssen & Johnson vaccine that also uses the pre-fusion stabilized S2P spike. They use a adeno 26 vector to deliver this vaccine. And one of the really cool things about their phase 3 clinical trial is that they are testing a 1-dose regimen as opposed to 2-dose regimens like the other vaccine candidates. AstraZeneca is not using the prefusion stabilized spike, but is delivering the full spike via their CHAD vector, or chimpanzee adenovirus vector, with about 40,000 participants in the United States and other large-scale trials across the countries. And then lastly, but certainly not least, is Novavax, that is using prefusion stabilized spike nanoparticle in combination with their adjuvant Matrix M, with over 15,000 people already enrolled in the UK and their US phase 3 trial to begin soon. All of the data that I showed to you today is synonymous across most of these vaccines, in that many of them are eliciting high-level efficacy in small animal models and non-human primates, eliciting robust neutralizing antibody responses, and the front runners are expected to have full efficacy results soon. And as you may have heard on the news yesterday, Pfizer-BioNTech's prefusion spike being delivered by mRNA has shown to be preliminarily 90% efficacious in their interim analysis of their phase 3 clinical trial. And so what does that exactly mean? These trials are randomized, placebo-controlled phase 3 clinical trial. There's a vaccine group, then there's a placebo group, and the volunteers know anyone associated with the trial, know whether you are in the vaccine group or the placebo group. The volunteers are closely monitored as they go about their normal daily lives. And then as COVID cases are diagnosed by the trial physicians, they're tallied. And the way that you get vaccine efficacy is that you tally more COVID cases in the placebo group than the vaccine group. So if you take the Pfizer-BioNTech example, they get about 90% reduction in COVID cases or disease in the vaccine group in comparison to the placebo group. Here is just a slide that I want to acknowledge everyone on the team at the Vaccine Research Center who's been working with me for several years. The COVID-19 response out of the VRC has been a VRC-wide effort. I'm probably leaving off names, but all of these people on this slide have been extremely helpful in various ways with deploying this Moderna vaccine candidate and then with other efforts from the VRC. We've collaborated very heavily with many laboratories across the United States. UT Austin and Scripps must be named because they started with us with some of our structure studies that led to all of this work, and also Moderna, which is manufacturing mRNA 1273. And the advancement of vaccines has been overall and across the world a multi-institutional effort. And so here's a couple of logos from various partners, whether it be from pharma or nonprofit institutions that are driving forward with driving these vaccine efforts forward. And with that, I will take some questions.
Kizzmekia, thank you so much. It's such a timely presentation. As you pointed out, with yesterday's announcement of an interim analysis that appeared to give 90% efficacy, why was the expectation for vaccine efficacy against this novel coronavirus so low?
Well, I think that, so I'm assuming that you're reading that people expected like a 70% or, right? Because this is literally a first generation vaccine. I remember like back in the day when I was younger and I saw the Gardasil or HPV vaccine results, and I was just like, there's no way a vaccine could ever be that effective. So, 70% and 80%, what people expected is actually okay, and what you would expect from a first generation vaccine that has, only been in trial for a year. And we didn't really know what to expect because we don't have the knowledge around correlates of protection and all of these things. So I think that as we stated our expectations, we were being fairly modest. And also these mRNA candidates in particular have not gone into phase 3 clinical trials enough to us to know how they might work. But I cried when I saw 90%. So I'm extremely thrilled about what it means around the entire vaccine portfolio. And essentially, I think probably the take home message is that if you're eliciting really robust antibody responses that you are probably going to have a good efficacy for these vaccines.
One of the students wants to know why we test so many different doses of vaccines. Why do we explore the lower doses so extensively? Why don't you just go to a very high dose right away?
So that is such a layered question. I like to think of myself as an academic, so I'll start on that side. So from an academic standpoint, testing very low doses allows you to assess breakthrough. So blowing out the response can actually give you a false hope around the vaccine. But when you go down in your doses and you start to see, for example, at that 0.01 microgram dose where we saw breakthrough, but the mice didn't die and they still recovered, that's actually very gratifying as you are in the preclinical stages. Because what that tells you is that you have a very efficacious vaccine and that you're not eliciting any bad responses by having subprotection, which subprotection might come up later in, nature or what have you. And then from a manufacturability and more on the pharma side, you want to test a lot of doses and pare your dosing down, because if you can give 50 micrograms instead of 100 micrograms, you can vaccinate double the amount of people across the world. Or 30 micrograms, as I believe Pfizer is doing, as opposed to the 100 of Moderna, the vaccine responses will be about the same, right? Because there's this plateau, but you'll be able to vaccinate 3 times as more people. And so there is also the business aspect to it.
As you pointed out, there's tremendous advantage to developing multiple vaccines simultaneously. But in the future, should the mRNA vaccines prove to be safe, do you see them as basically taking over in the way we develop vaccines against pathogens in the future?
I think that it is going to be hard. Shall the mRNA vaccines prove to be eventually completely effective after the phase 3s? And when we look at durability, with the timeline that these companies have been able to develop these vaccines in a pandemic response, I think that it's going to be very difficult to convince people to develop other types of vaccines. But other types of vaccines in the interim come in handy, because mRNA vaccines have to be stored at -80. So getting them to the developed world is going to be difficult. You probably won't be able to use mRNA vaccines in maybe in certain types of populations where other vaccines might be able to use. So there are still holes. And technologies, will continuously get better. I just don't know how you're going to get better, at least as far as I can tell. I think that what will happen with the other platforms is they will start to fill in some of the gaps. And there will be instances where T cell responses, for example, CD8 T cell responses are more important for protection for different viruses. And in that case, an advector might be better.
You made an elegant case for knowing a lot about the structure of a protein before you decide exactly what to put into a vaccine of this type. Do you see in the future adding additional components of this coronavirus to optimize its efficacy?
Probably not for optimizing its efficacy. But what I do see is optimizing its other things, like its breadth, for example. So, the NA, not the NA, I'm not in flu world, I'm in coronavirus world. The N protein is more conserved across the coronavirus family than the spike protein. You might not get neutralizing antibody responses, but as we learn more about the types of responses that might be broad, I think that people probably will monopolize on using different types of proteins.
And there's some curiosity about the steps that the FDA is making, or that companies are making, or the government, to ensure that people of different ages, races, et cetera, are all being tested in these trials.
Can't speak for the regulatory agency. I am not an employee of the FDA. I am an employee of the National Institutes of Health and a close collaborator with one pharmaceutical company, Moderna. So I can speak from our perspective. we were very, 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 very keen on making sure that there was ethnic diversity in the phase 3 clinical trial. Actually, it slowed up our phase 3 clinical trial because the CEO of Moderna made a statement that we would not enroll essentially what are white men anymore and start enrolling more people from Latino or African American backgrounds, et cetera. And so in the phase 1 clinical trial, we had about 4% minority or something around that way. And then eventually in the phase 3 clinical trial, we have like over 30%, 10% of which is African American. So the phase 3 clinical trial demographics mirror what you see in the US population, which is what you want for a vaccine trial. And I'm very happy with the results of that type of enrollment. I do know that I'm not sure how that affects regulation around who can get the vaccine. I think probably the FDA has to figure that out. But to have that type of broad enrollment in your phase 3 and to be able to say that your safety and efficacy signals are generalizable across populations is a big deal.
Well, with that, I want to thank you. It does seem like there's a light at the end of the tunnel, and we so appreciate you educating us on this vaccine and your work in it. Thanks so much, Kizzmekia.
Thanks a lot for having me. This was fun. I never really get to talk broad, so it's very fun to be here. Bye, everybody.